got my hands wet in the lab, working on making of different diagnostic growth media, taking bacteria and culturing it, growing it in the petri dish and all of that, and starting to see how these laboratory tests were in action, how they were used. We could arrive at a conclusion that we were looking at the group A beta hemolytic streptococci, and so I was very excited that I could identify this organism in that pathway. So it got me in the lab, it got me exposed to that kind of thing, got me exposed to record keeping, and when it came time for me to go to a science meeting, I realized that all that information I had been recording was actually going to be analyzed in ways that I had never thought of and didn't understand, but she could make sense of it. She was telling me how to group these different conditions and all of that, and before too long, we started to see trends that came out of numbers that I thought were just numbers. Didn't see that, and so I became very interested in how science worked after that. I wanted to understand not only that I was doing this work, but also how to understand what the data meant. And when I could see it like that, and then talk about it in some authoritative way through a poster or through a presentation, which I did both actually. I was very happy to do that, and it took me to places that I had never been outside of my reservation. Like I went to Atlanta, Georgia, I went to Washington, D.C., and other places to talk about my research. And all of that was a whole new world for me. It was totally different than anything I'd ever experienced coming from the reservation. I didn't really know exactly what I wanted to be. I liked microbiology, that's what I worked on, but again, technology came to my doorstep at Denetclaw College. The microscope salesman came by and showed us a fluorescence microscope to help us with our research on identifying the group A beta hemolytic streptococci. And I didn't know what that was, but under the first illumination of this organism under fluorescence excitation and seeing these beautiful fluorescent colors, you can just think of the movie Avatar. It was just like that. The whole world just opened up. I never thought I would see things in that kind of color before. And for me, that was it. I mean, I was going to be a cell biologist, and I was going to use fluorescence in my research.